Hey everybody, this is Daylon and Arun, and we're the hosts of the STEMCELL podcast. Welcome back to day 2 of our video series, rounding up the latest research presented at the 2020 ISSCR virtual meeting. In addition to these videos, don't forget to be on the lookout for our upcoming ISSCR episodes, where we'll be speaking with some of the meeting's top researchers, including Drs. Madeline Lancaster, Christy Redhorse, and Martin Pera. You can stream the episodes at www.stemcellpodcast.com or download them wherever you get your podcasts. We've got some interesting research from the meeting that we want to highlight today, but before we get to that, Arun's got a brief message for you guys.
Yeah, Stem Cell Technologies is giving an innovation showcase on the applications of organoid and organotypic cultures in infectious diseases, nephrotoxicity, and highly relevant cell-based assay development. That's a mouthful. With a live presentation and Q&A at the 1 p.m. Eastern Time tomorrow. So you can also check out the poster session for some more data from the scientists behind the products. You can also find the full schedule for live sessions at STEMCELL's virtual booth.
All right, so, we're recording these about midday for the conference. There's all these morning and early afternoon sessions, and then they go deep into the night, some nights. So we're going to catch you up on what you missed from yesterday. That was day 1 in the afternoon. There were a few sessions. First one that I wanted to talk about was stem cells as drugs in clinical trials. I thought this was there were multiple sessions and we can't cover them all. But I want to highlight this one because it was packed with some really high-level clinical translational stem cell researchers. It was headlined by Agnete Kirkeby at Lund, who was looking at Parkinson's disease and engraftment of pluripotent stem cell-derived dopaminergic neurons in the context of Parkinson's, high-level stuff. And then we had David Williams from Boston Children's Hospital talking about gene editing for sickle cell anemia. And then the crowning it was Sonia Schrepfer from UCSF, and she was talking about these immunogenic iPS cells and their derivatives that have made her famous. I think the real thing for me here, Arun, that was so great about that session was that it showed how far we've come. I've been in the game now a couple decades, and I remember early days in the first conferences you'd go and someone would present that they got their dopaminergic cells to survive in the brain, and people would stand up and cheer. And here you had Dr. Kirkeby showing some amazing work, moving the work towards the clinical trial, and showing that you could not only engraft these things and get them to survive and proliferate and have longevity, but they would actually have projections from one end of the rat brain to the other. So really amazing progression of the work. And by the same token, David Williams was talking about the gene therapy and how far we've come with sickle cell anemia. We've been talking about gene therapy now for multiple decades, going on maybe 40 years. And it's been more of a challenge than I think we expected. Maybe a harbinger of what we can expect with stem cells there. But now we're getting there with sickle cell and this new gene therapy that he talked about. Also, Jack Heath from Editus had a follow-up talk today about that. So you can check those out on demand. And lastly, Sonia Schrepfer, although, I think there's still a lot of challenges standing in the face of hypoimmunogenic cells, basically the fact that the immune system doesn't see them. Challenges there, but still amazing to see how you had all these technical roadblocks that were set up in early days that now are falling just by the ingenuity of the scientists out there. And just on the flip side of that, Arun, quickly, I'd also like to say that I was kind of a little bit, I wouldn't say frustrated because I'm not the one doing the work, but I was a little bit overwhelmed by how much is necessary, how much, preclinical work needs to be done and the quality controls, et cetera. Lots and lots, tens of things that you need to do in order to even make these things eligible. So while it was also really inspirational in terms of the progress, it really made me realize that getting cells into people is it's regulatory wise, it's a major challenge, notwithstanding, or not to mention the biological challenges out there. The regulatory hurdles are very high.
Yeah, absolutely. Like you mentioned, back in the day, it was just a matter of getting these cells to integrate into the portion of the body that you were interested in studying, whether it's the brain or, recently people have even used, iPS-derived pancreatic beta cells as also another form of cellular therapy. These cells are able to generate insulin. There's companies like Sema Therapeutics who are working on stuff like that. So that's something that's also in the work. Editas, of course, we've heard a lot about Editas when it comes to bringing genome editing and CRISPR to the clinic. I don't think they actually talked about this in this particular talk, but some of their initial work is using CRISPR in the eye, and that's been pretty effective from everything that I've heard, at least on the preclinical side of things. But you mentioned about preclinical and how you were a little bit disheartened about how tough it is to actually make the jump from the preclinical to the clinical side of things. But I'm maybe I'm conservative, but I definitely think that's the way to go. We got to be careful when it comes to actually bringing these things into the clinic, right? Especially when it comes to cell therapy. I think small molecules or even antibodies is one example. But when we're talking about a live cell, which has issues when it comes to integration, immunogenicity, even potentially differentiation, I think these are all things that we really need to carefully consider on the preclinical setting before we even think about making that jump to the next step.
Yeah, I think you're absolutely right there, and I don't mean to say that we should rush, of course, but I just think that it's forced a lot of people, I think, to reframe over the course of all these years. I think we've had to reframe our expectations, and I think what we're realizing is, as you alluded to it, is that this isn't like biologics or pharma, drugs, compounds. This is a live cell product. And the FDA is learning along with the scientists and what they need to do and what kind of regulatory, I wouldn't say obstacles are barriers, but I think thresholds is the word. I think that the FDA, and I think also not to go deeper down the rabbit hole, but there was, one thing, Arun, I will say about the virtual format that's amazing is that I couldn't sleep in the night, so I just went back and on demand saw everything that there was to see. So I'm totally caught up in the conference. I don't know if I'm going to be able to stay up to speed with all the concurrence going on, but I looked into some other sessions, and there was one on, funding and the industrial apparatus, regulatory apparatus, and approvals. You're getting a lot of information if you go to all these talks about how involved it is in the details. And, that's really what it's about, is the details. I think it was nice to talk about the ideas and the potentials and the hopes. But now that we're talking about the reality, reality is about details. So we've got a long way to go, but we've come so far.
The devil is in the details after all. And, on the note of you staying up late at night to watch these things, these presentations are available for 30 days after the conference ends. So it doesn't matter what time of night, morning, afternoon, whenever you watch them, they'll be around for a month. So, if you can't catch the plenary session or can't catch, your favorite workshop from your favorite speaker, don't fret. You can come back and take a listen to it. So I guess we can move on to this morning's session. So this is Wednesday, U.S. time, noon, close to 1 o'clock on the West Coast here in L.A. So we're actually recording right before the major first plenary session, the presidential symposium. But we're going to recap some of the stuff that we thought was really exciting from this morning session. And there are a couple of big, groups of talks that we both listened into. The first was a science advocacy and communications seminar. All right. I thought this was great because this wasn't, really hardcore nitty gritty science. We're getting into that in the sessions to come, like this afternoon. But this was really enlightening to me because it was almost like a story time. It was story time from some of the luminaries in our field, like George Daley, for example, who's now the head of the Harvard Medical School, the dean of HMS, and also a longtime hematopoietic stem cell researcher. He was talking about his journey to and from Capitol Hill here in the United States, trying to pitch stem cell biology to these non-scientist politicians, and what that process was like. He gave a quip about some people didn't know the difference between implantation and ingestion of
No.
embryos.
Yeah.
Yeah, I'm sure you caught that. That was so. Hey, that's more power to you. You know, part of what you have to do, at least in my opinion, as a scientist who's funded by taxpayer money, is try to advocate for your science. And I think George Daley has been the guy, one of the guys for advocating stem cell biology in the United States.
Yeah, I think it was, he said that Dianne Feinstein said, so what happens if you ingest these embryos?
Well, a
Yeah,
lot can happen.
that was it was, like you said, it was story time. It was like an origin story. And the takeaway because I didn't know that. Sean Morrison also spoke, and he said a similar thing. In his case, it was that he was, they were asking him to weigh in on this referendum that they were going to try and write up to try and quell all the anxiety of all the rabble rousers out there who were talking about cloning and cow-human hybrids and whatnot. And he looked at the doc and was like, well, this is just not going to do the trick. And so he stayed up all night and rewrote the doc. And so by rewriting the doc, just by anticipating effectively all their arguments and shutting them down preemptively, he became the kind of de facto advocate and the one that people look to because he was the guy that took the initiative there in the middle of the night while I'm listening to ISSCR videos. With my eyes all burnt out. He's writing policy, right? And George Daley was a similar thing, not the same, but similar in that there was all the hysteria, they called the Whitehead, and he was the guy there who was working on embryonic stem cells in the mouse, and I'm sure applying his interest to the human as well in hematopoiesis. But he was the guy, and he was the guy that got put on the phone and thereafter became the routine visits to Washington to testify before Congress. So it kind of just tells you that you become that guy just by doing the thing. Take the initiative. I feel lucky that I stumbled in this podcast. I'm not trying to put myself on par with the science communicators and advocates like that, but this is a great opportunity, right, Arun? We get to talk to all these people. We get all this access. We get to just enjoy our science. And it's a privilege that kind of knocked on our door, I think. So you get the privilege by doing the thing and taking the initiative, and sometimes you just get lucky and you're the one who picks up the phone, I guess.
Yeah, no, we're definitely quite fortunate. I think as a member of the, I guess, so-called post-IPSC generation, you kind of take these things for granted when you think about how tricky stem cell biology and human embryonic stem cell research was back in the day. I mean, Dr. Daley talked about the transition from the Bush administration to the Obama administration and the various restrictions that were in place during that time, right? I think another quote that he said that stuck with me was that advocacy takes time, and I think that's very true. His journey is a case example of that. Advocacy truly takes time, and it's not until now that we have so many more freedoms when it comes to actually doing stem cell biology and stem cell research. One other thing that he mentioned was that some people and some scientists actually view advocacy as a distraction from the science. And I'm of the opinion, of course, that that's not the case, and these two things go hand in hand. I think we have a responsibility to the public, as I mentioned, since we are funded by public money, and I think Dr. Daley agrees with that particular perspective.
Yeah, absolutely. I think there's a lot of archetypal ideas about science that are changing. There's the lone scientist, siloed up, and that's changing. It's much more collaborative. And in the same vein, it's this idea of are you doing your basic in the lab? It's for the pursuit of knowledge versus we're making products to serve humankind, right? And paid by the taxpayers, and they're funding this work because they want to benefit at the back end. And part of that is commercialization. Just to go back quickly to Mar, because I forgot to mention another big splash for me and one of my favorite speakers. And in this case, she really demystified that whole commercialization process with Sangeeta Bhatia at MIT. She spoke in the what was it called? Stories from the Frontline session. You guys should check her out at least. There was a couple other speakers in there. One of the acolytes of Gordana, like Casey Ronaldson, who we had the show, Ick, who had started Epibone and others. But Sangeeta, she really, really took the audience through her journey, starting these 3 companies, and talked about how each of them were different, like her children, but like children, she had to step out from the authority and role and move on as a scientist and let them kind of take shape. And it was for me really interesting, fascinating, and demystifying, because it's always hard for me to see the line between academic scientist and the head of the scientific advisory board, or the CSO, or whatever, or the founder. But when you hear her talk about it, it seems like the most natural thing in the world. So guys, I would recommend you check that out as well, because she's just such a great speaker, a great scientist, and an inspiration.
Yeah, she's absolutely tremendous. She is one of the greatest stem cell bioengineers that we have on the planet. Of course, she also benefited from being mentored by Dr. Bob Langer, who is also over there in Boston. You can wake up in the morning and get a patent. That's kind of what it seems like over there, right? Patents just left and right. But she's an absolutely tremendous scientist and an inspiration to a lot of the people in my generation. Other people that we were really excited to listen to included Fiona Watt, also from the Advocacy seminar. She's over there at King's College London. She was actually talking a little bit more on the British side of things in terms of how advocacy plays out over there in the United Kingdom. She actually had a couple of important quotes as well. So when it comes to actually engaging with members of the public, she likes to ask herself, what is the message that I actually want to convey? And also you have to really consider the importance of your target audience. If you're going to engage with funders, it's obviously a very different pitch that you're going to present, as opposed to if you engage with students or politicians, right? So she mentioned using simple language is also really important, but don't underestimate the intelligence of your audience. Not every audience is as knowledgeable about stem cell biology as the audience of this podcast, but the people out there, they know what they're talking about, especially if they have an interest in the science, even if they are general public. Don't underestimate the intelligence of your audience.
Yeah, her final piece of advice had me a little bit shook, because she said, you shouldn't make off-the-cuff remarks. And this is not
That's
maybe,
a problem for us.
That's not my strong suit
for you,
there.
maybe.
Well, I'm working on it, all right? I'm keeping the remarks on the cuff, maybe starting after this episode. I don't know, maybe. So yeah, that was great. And then also, of course, the Godfather, George Church, he swooped in on the organoid session, but I don't know that he added so much science to that, but he just added an air of some gravity to it, like Gandalf in the Lord of the Rings. But I think the highlight of that session, I don't know, Arun, I think you're going to agree, was the next level stuff from Serge about the organoids, the assembloids, as he called them, Sergio Pasca. Let's talk about it, man. I mean, this was, I think, the highlight of the day for me.
That was unbelievable stuff. And the other thing that was really exciting to me about that particular talk was that was all unpublished data that he was presenting, So yeah, we can just cut right to the chase. Basically, his claim to fame and Sergio Pasca's lab's claim to fame is the generation of these so-called, organoids from different portions of the brain. So cerebral organoids, ventral forebrain organoids, dorsal forebrain organoids. But lately they've shifted to these so-called assembloids, where you're basically putting together multiple organoids so that you can have like a systemic approach, a systemic platform to understand these complex biological processes, right? And so this amazing example of an assembloid was a corticomotor assembloid, whereby you're actually connecting a cortical organoid to a spinal organoid to a muscular organoid. And not only that, and this was the mind-blowing part of it, you could stimulate the cortical organoid, and then you would have a response, a functional response in the muscular organoid. It would actually start contracting. And they were able to do this repeatedly so that the muscular organoids were able to mature over the course of like hundreds of days. This is like science fiction stuff, man. That was really just unbelievable work that they're doing. I can't wait to see the paper.
Yeah, it's probably coming out any day. And I mean, like you said, it was so next level It's like, why stop at compound organoid when you can make like a triple organoid? Yeah, of course. And then why stop at like, yeah, just putting it together and doing the gating? We'll do a little optogenetics in there. Yeah, sure. Why not? Why wouldn't we? And then they hooked it up to the fetal limb from a mouse just for the goof, because that's like more physiological, and so is inducing twitching of that. And then the skeletal muscle organoid. So yeah, this was next. And when you start to go to that, I mean, I don't want to overstate it, but if it's, two is, you know what they say, twice, I mean, once is never, twice is always. I think that here is like one organoid was one thing, and when you had the compounds, it was like, okay, you can put two things together. When you have three things, you're like assembling a tissue pretty much. So I think that this is a real breakthrough conceptually. Because those three components as well are so essential to the unit, and there's such diverse elements of the unit that all work together in this circuit. So like you said, I can't wait to see where that paper comes out and some of the details, because he had to talk about all that in like 10 minutes, which was shortchanging him. But, the session is the session, so we'll have to wait for the print.
Well, the good news is actually we're going to be having him on the podcast not too long from now. So he is going to be a guest on an upcoming episode of the podcast. So stay tuned for that.
Get out of here. Wow. I didn't even know that. Arun, you must have heard that on Twitter. I'm sleeping.
Breaking news.
While we're talking about the podcast, I got a message quickly from STEMCELL Technologies. It's a neural hematopoiesis connection here. We're talking. I'm advising you to check out this upcoming product from STEMCELL Technologies. It's StemDiff microglia media kits. They're used to differentiate HPSC-derived microglia from hematopoietic progenitor cells for modeling neuroinflammation, neurodegeneration, and co-culture applications. You can sign up now to be the first to know when it's available at HTTPS. I don't know why we put that in there. www.stemcell.com/ISSCR 2020 FTK. I think it's appropriate because we're talking not only about neural here and the neural organoids section and the ethics of organoids, which we're going to get to in a second, but Sergio Pasca, he's doing this compound thing, and I love hematopoiesis. I love the idea that you need hematopoietic cells for the brain to work. All right, you need me, neural. I don't need you. But let's move on. The ethics session. We had Alice on Watry on the podcast. That was a big episode for us, a highlight, I think. And one of the things that was really notable about that is just the concept of these organoids starting to have brain waves that were similar to fetal brain waves. And I just want to jump right to one minor note that was slipped in by, the Japanese researcher, ethicist Misao Fujita. She was talking about the series of all the organoid articles, which of course are escalating, accelerating in recent years. But she just dropped in quickly, pointed to the Moatri article and described it as potentially a, quote, trigger for awareness. And I took that as maybe a euphemism or an allusion to the fact that I think now this whole idea of consciousness, I guess, you want to call it brainwaves, however you want to extrapolate that whole notion is gaining steam in the popular media and is kind of bearing a lot of scrutiny in terms of the ethics of that. Arun, what did you think about those components of the session?
Yeah, no, I think it was fantastic. I thought it was a really well balanced session because you have the scientists who are pioneering this cutting edge biology, like Sergio Pasca and also Paulo Arloda, for example. They're pioneering this biology, but the reality is, if we're actually thinking about consciousness and we're thinking about the true power and efficiency of these organoids and whether they're actually comparable to the real deal, they're pretty far from it, right? In fact, George Church had a slide comparing the real human supercomputer, the real human processor in the brain, and to see how efficient that is in comparison to other so-called processors. Like the big one is a silicon processor that we use on our computers, right? And it's still orders of magnitude when it comes to the biological processor, orders of magnitude more efficient and more advanced. So I think we still have a very long way when it comes to actual true consciousness, but that's the fun part, right? That's the fun part, is that we can actually have these discussions, combining input from not only the basic scientists, but also some other very high profile bioethicists who were on this talk. In fact, the moderator was Insu Hyun, who is very well known when it comes to the bioethical analyses of different stem cell based technologies. Also, Jintine Lunshoff was another bioethicist on the call. I think one thing that she mentioned that really stuck with me, a couple things. One is that a lot of people run with the idea of these so-called organoids and call them brains in a vat. And she said that was entirely irresponsible to call them that from a scientific perspective and also from a public's perspective. And I agree. I think we have to call them for what they actually are. I know it's a sexy phrase to actually call them a brain in a vat, but it's so far from that. And the other thing was, she said that when it comes to what brain organoid researchers have to be most concerned about in the near term when it comes to actually disseminating their work, she said there was three things you have to consider. One is hype, the hype associated with your work, the hope that it can potentially bring to people afflicted with different neurological conditions, and hubris, right? Hubris is something that's also really important because, of course, as scientists, we do want to be, a lot of times we want to be first, and we want to be the ones who are known to have the claim to fame to a particular technology. But we have to take it slow and take these things into consideration, especially when you're talking about such a charged subject as human consciousness that potentially you might be developing in a dish.
Yeah, I remember she said that the counterpoint to hype/hope is self-deception. Right. And the hype part of that can really have a negative effect on the public's perception. And I think Karen Youngsma, who came after Jantine, she kind of addressed that, talking about a survey that they took of the patient perspective. And I thought that was really interesting because, we're all well versed on when someone says a brain in a dish, as a scientist, our first inclination is to say, it's not an actual brain. So know that it's language that is sexified in order to talk about what it actually is, which is a deconstructed cellular apparatus that maybe is in the neural lineage in 3D. Sure. And although we're getting closer with these Pasca compound assembloids. But I think the point there that really was driven home for me was that when you surveyed patients and you said, what's a brain organoid? They were a total blank slate. They're like, brain organoid? And then their imagination just took over. And she had this nice slide where it was a picture of a sponge on one side. So some patients just like, brain, little, maybe looks like a little sponge. And the other side is a picture of Terminator. So that's, I think, the dichotomy that you have in these patients is that you leave this void for their imaginations to fill in. And a lot of that's going to be filled in with, I think, a cynical worldview, especially given the kind of abuses that people have seen in the media, that are science related. I'm talking about the whole gene editing fiasco. I'm talking about these mesenchymal stem cell clinics. So we don't want to fan the flames of that distrust by having these, these images that we plant in there. She said that the fears, and of course everyone's on board for science. When you talk and explain what's good about it, they're willing to donate. And by the way, this is in Europe, where I think they're probably much less cynical than here. Right. But the fears that they had were, one, that they would devalue the uniqueness of humankind. Two, the misuse, a la like Terminator. And three, and this is so huge, what it would be like for the organoid. And that's what we're talking about. When we start talking about brainwaves and consciousness, I think it's what these ethicists and scientists are really starting to wrestle with, is that the public, as soon as you say brain in a vat, they're like, oh man, that poor brain. What a miserable existence. And the one that said, the slide said, the brain can't even vocalize that it wants to die. Whether, no, sorry, I should correct that. Whether it wants to live or die.
Right.
because of course it doesn't have a mouth. And I think that, I mean, when you explain it to somebody, you said, speak to like you're talking to a 14-year-old, is what Fiona Watts said. And I think even if you explain what a brain organoid is to a 14-year-old, they're not going to go thinking, oh, that poor organoid, it probably is suicidal.
Yeah.
But when you just say brain in a vat, like you said, I mean, you're leaving it up to them to fill in the gaps there. So we got to be careful.
Hey, you know, that's, like I said, that's part of the reason that I think stem cell biology is just so exciting, is because we can have conversations like this, right? What other field? I mean, there's a few other fields in biology where, you know, that are really at this cutting edge of bioethical debate. And stem cell biology, from its inception, has been on that cutting edge. It's fun to be a part of, you know?
Yeah, it is fun. And you know what else is fun? Doing these videos. I want to do one every day forever. But that's it for this one. That brings us to the end of today's video. Make sure to follow us on Twitter at STEMCELL Podcast to find out what we're doing at ISSCR 2020. And check back here tomorrow for another video roundup of all the interesting research that's taken place over the previous 24 hours. So that's the afternoon sessions. There's this late session with my man, Tamashima, although it's late. I'm going to stay up for that, Mark. All right, so you better be good. I'll tell you about that tomorrow. And tomorrow, we're going to talk about these plenaries too, right? So it's on.
Yeah.
And we got some concurrence in there maybe too. So it's going to be a full day. We're going to have to pick our best of, but it shouldn't be hard. Well, it will be hard, but it will all be good for sure. Join us for that, guys. We'll see you tomorrow. Thanks for listening and watching.